For this video, I will be discussing the connections between iron deficiency anemia and type 2 diabetes. This topic is important to discuss as there is a high prevalence of both diseases. Iron deficiency anemia, according to the World Health Organization, is the top nutritional disorder in the world. Research suggests that as many as 80% of people in the world don't have enough iron in their bodies. It also suggests that as many as 30% of people have anemia due to prolonged iron deficiency. According to the CDC, type 2 diabetes is prevalent in more than 34 million Americans, which is 1 in 10, and approximately 90 to 95% of them have type 2 diabetes. And globally, an estimated 462 million individuals are affected by type 2 diabetes, which is corresponding to 6.28% of the world's population. So these two images are visual representations of the prevalence of each. So on the left is the prevalence of anemia worldwide, and on the right it's the prevalence of type 2 diabetes worldwide. The research question I will be exploring today is, can iron deficiency and type 2 diabetes be caused by genetic susceptibility due to TMPRSS6? Type 2 diabetes is a chronic condition where the regulation of blood glucose levels are hindered and usually develops over time due to diet-related reasons. Iron deficiency and iron deficiency anemia negatively impact glucose homeostasis and glycemic control, which is a problem for homeostasis and the likelihood of type 2 diabetes, as glucose and insulin work together to help regulate blood sugar levels. Anemia is when your body is not able to carry enough oxygen to tissues because of a low amount of healthy red blood cells. ACTA Biomed research concluded diabetes can contribute to anemia through reducing absorption of iron, gastrointestinal bleeding, and through diabetic complications that cause anemia. IgA and increased A1C levels are correlated, meaning when iron stores are reduced, glycation of hemoglobin A1C increases. From Iran Biomed research, this leads to false high values of HbA1c in non-diabetic individuals. HbA1c is the most important predominant fraction of HbA1, and it is formed by the glycation of terminal valine at the beta chain of hemoglobin. It reflects the patient's glycemic status over the previous 3 months. Because of this, it makes sense that ID and IgA is seen more commonly in those with diabetes than those non-diabetic. The TMPRSS6 gene codes for the protein matriptase 2, a serine protease which functions as the negative regulatory protein of hepcidin transcription. Hepcidin is an iron regulatory hormone. Serine proteases hydrolyze proteins and play an important role in catalysis. This image is a representation showing the role of TMPRSS regulation. Matriptase 2, also known as TMPRSS6, is a critical regulatory hepcidin in the liver. Matriptase 2 cleaves hemojuvelin, which is involved with iron absorption and iron homeostasis regulation. This subsequently affects the signaling of bone morphogenic protein, or BMP, that promotes bone growth and healing, and the signaling pathway which activates hepcidin. This image is showing the discovery and development of TMPRSS6 inhibitors regulating hepcidin levels. Iron refractory iron deficiency anemia, also known as IRIDA, is a source of hereditary anemia. This is caused by mutations in the TMPRSS6 gene that maps chromosomes and encodes matropase 2. This disorder was discovered after research linked hepcidin as an essential regulator of iron homeostasis. When the body lacks the sufficient amount of iron, this leads to low red blood cell counts that are needed for the delivery of oxygen and tissues in the body. On the other hand, genes can also cause iron levels to be too high. Autosomal recessive disorders means that one mutated gene is inherited from each parent. Hereditary hemochromatosis is an autosomal recessive disorder that disorganizes iron balance by exceeding the proper iron absorption, accumulation, and parenchymal cells, which are the tissues that help organs perform their biological function. There are subordinate types of HH disease, and each occur due to mutations in the HFE, TFR2, and HJV genes that codes for hemoglobin, which has an anti-inflammatory effects when iron gets too high, or the gene HAMP, or hepcidin. HFE, TFR2, and HJV each contribute to hepcidin regulation. The inheritance of iron deficiency due to the TMPRSS6 gene is rare, but possible through recessive genes. It is also important to be able to differentiate between IRIDA and nutritional iron deficiency. For example, if IRIDA is present in 3 siblings, it is highly likely that their cases are due to inheritance. Although, for a number of IRIDA patients, the cases occur at irregular intervals. This can happen if there's a small pedigree size and if recessive transmission took place. Clinical data is generally the method used to effectively determine if iron deficiency is inherited or required. Loss of blood or decreased iron absorption are indicators of iron deficiency that's been acquired. Inherited transmission is usually detection in childhood through routine blood tests. Another important difference to note is iron deficiency versus iron deficiency anemia. One indicator that differentiates iron deficiency anemia is red blood cells, which are usually higher, while serum deficiency is lower in iron deficiency. Serum ferritin is a protein that contains iron and is useful for being able to determine how much iron your body is storing. Published by Hematologica Journal, overall screenings for mutations based on the sequencing of exons and exon intron boundaries of the TMPRSS6 gene is the most common technique when testing IRIDA patients. Like mentioned before, those that have anemia have less red blood cells than those that do not have anemia. And along with that, the red blood cells that they do have are usually in unusual shape, or that are larger than normal or smaller than normal. And these differences can be seen in this image. For my recommendations and conclusion, based on the research and studies conducted, it is well suggested that iron deficiency, a symptom of type 2 diabetes, can be due to genetic susceptibility by mutations in the TMPRSS6 gene. In an attempt to combat iron deficiency with the genetic susceptibility already in the body, taking iron supplements and vitamins like vitamin C will help increase iron absorption and thus iron levels. HbA1c levels can be reduced through iron therapy too, along with eating carbohydrate-rich and whole foods. And that's all. Thank you for watching.